This article is about a biological genus. See Ebolavirus for the specific species within this genus responsible for the 2014 outbreak. The genus Ebolavirus is a virological taxon included in the family Filoviridae, order Mononegavirales. The species in this genus are called ebolaviruses. 5 species are known, and 4 of these cause Ebola virus disease in humans, a type of hemorrhagic fever having a very high case fatality rate. The 5 known virus species are named for the region where each was originally identified: Bundibugyo Ebolavirus, Reston Ebolavirus, Sudan Ebolavirus, Taï Forest Ebolavirus and Zaire Ebolavirus. The Zaire Ebolavirus species is the type species for Ebola virus. It has a sole known member, which since 2010 has been called simply Ebola virus. This virus has the highest mortality rate of the Ebola viruses and is also responsible for the largest number of outbreaks of the 5 known members of the genus, including both the first documented outbreak and the outbreak with the most deaths. The name Ebola virus is derived from the Ebola River in Zaire, where Ebola virus was first discovered, and the taxonomic suffix virus. This genus was introduced in 1998 as the Ebola-like viruses. In 2002 the name was changed to Ebolavirus and in 2010, the genus was amended. The genus Ebolavirus is closely related to the Marburg virus. Taxonomy notes, Ebolavirus is pronounced. According to the rules for taxon naming established by the International Committee on Taxonomy of Viruses, the name of the genus Ebolavirus is always to be capitalized, italicized, never abbreviated, and to be preceded by the word genus. The names of its members are to be written in lower case, are not italicized, and used without articles. Genus inclusion criteria: a virus of the family Filoviridae is a member of the genus Ebolavirus if its genome has several gene overlaps, its 4th gene encodes 4 proteins using co-transcriptional editing to express SSGP and GP1, 2 and proteolytic cleavage to express SGP and dipeptide. Peak infectivity of its virions is associated with particles of ml 805 nm in length. Its genome differs from that of Marburg virus by a per-mil 50N and from that of Ebola virus by less than 50% at the nucleotide level. Its virions show almost no antigenic cross-reactivity with Marburg virions. Classification: the general Ebola virus and Marburg virus were originally classified as the species of the now obsolete Filovirus genus. In March 1998, the vertebrate virus subcommittee proposed in the International Committee on Taxonomy of Viruses to change the Filovirus genus to the Filoviridae family with 2 specific genera, Ebola-like viruses and Marburg-like viruses. This proposal was implemented in Washington, D.C., as of April 2001 and in Paris as of July 2002. In 2000, another proposal was made in Washington, D.C., to change the like viruses to virus, resulting in today's Ebola virus and Marburg virus. The 5 characterized Ebola species are: Ebolavirus Er, formerly known as Zaire virus, or Zaire Ebolavirus, has the highest case fatality rate, up to 90% in some epidemics, with an average case fatality rate of approximately 83% over 27 years. There have been more outbreaks of Ebola virus than of any other species. The first outbreak took place on August 26, 1976 in Yambuku. Mabel Lola Keller, a 44-year-old schoolteacher, became the first recorded case. The symptoms resembled malaria, and subsequent patients received quinine. Transmission has been attributed to reuse of unsterilized needles and close personal contact. The virus responsible for the 2014 West Africa Ebola virus outbreak, with the largest number of deaths to date. Sudan Ebolavirus Er, like the Ebola virus, SEBOV emerged in 1976. It was at first assumed to be identical with the Zaire species. SEBOV is believed to have broken out first amongst cotton factory workers in Nzara, Sudan, with the first case reported as a worker exposed to a potential natural reservoir. Scientists tested local animals and insects in response to this. However, none tested positive for the virus. The carrier is still unknown. The lack of barrier nursing facilitated the spread of the disease. The most recent outbreak occurred in August 2014. 13 cases were reported in Zaire, Democratic Republic of Congo. The average fatality rates for SEBOV were 54% in 1976, 68% in 1979, and 53% in 2000 and 2001. Reston Ebolavirus Er. This virus was discovered during an outbreak of simian hemorrhagic fever virus in crab-eating macaques from Reston laboratories in 1989. Since the initial outbreak in Reston, Virginia, it has since been found in non-human primates in Pennsylvania, Texas, and Siena, Italy. In each case, the affected animals had been imported from a facility in the Philippines, where the virus has also infected pigs. Despite its status as a Euro4 organism and its apparent pathogenicity in monkeys, EBOV did not cause disease in exposed human laboratory workers. Taï Ebolavirus, also referred to as Taï Ebolavirus and by the English place name, Ivory Coast, it was first discovered among chimpanzees from the Taï Forest in Côte, Africa, in 1994. Necropsies showed blood within the heart to be brown. No obvious marks were seen on the organs. And one necropsy displayed lungs filled with blood. Studies of tissues taken from the chimpanzees showed results similar to human cases during the 1976 Ebola outbreaks in Zaire and Sudan. As more dead chimpanzees were discovered, many tested positive for Ebola using molecular techniques. The source of the virus was believed to be the meat of infected western red colobus monkeys upon which the chimpanzees preyed. One of the scientists performing the necropsies on the infected chimpanzees contracted Ebola. She developed symptoms similar to those of dengue fever approximately a week after the necropsy, and was transported to Switzerland for treatment. She was discharged from hospital after 2 weeks and had fully recovered 6 weeks after the infection. Bundibugyo Ebolavirus. On November 24, 2007, the Uganda Ministry of Health confirmed an outbreak of Ebola in the Bundibugyo district. After confirmation of samples tested by the United States National Reference Laboratories and the CDC, the World Health Organization confirmed the presence of the new species. On February 20, 2008, the Uganda Ministry officially announced the end of the epidemic in Bundibugyo, with the last infected person discharged on January 8, 2008. An epidemiological study conducted by WHO and Uganda Ministry of Health scientists determined there were 116 confirmed and probable cases for new Ebola species, and that the outbreak had a mortality rate of 34%. Evolution. Rates of genetic change are 1/100th as fast as influenza A in humans, but on the same magnitude as those of hepatitis B. Extrapolating backwards using these rates indicates Ebola virus and Marburg virus diverged several thousand years ago. However, paleoviruses of filoviruses found in mammals indicate that the family itself is at least tens of millions of years old. Genus organization, table legend, denotes type species, research. A 2013 study isolated antibodies from fruit bats in Bangladesh against Ebola, Zaire and Reston viruses, thus identifying potential virus hosts and signs of the filoviruses in Asia. References. External links, ICTV files and discussions, discussion forum and file distribution for the International Committee on Taxonomy of Viruses.